this actually originated in when Daniel Nix, I think, was giving his presentation, and he had mentioned that the water department does PCR tests, and he just sort of said it in passing. And as I was thinking about it, it's like, well, maybe not everybody knows what that even is. And it is useful. We can use it in terms of, I mean, it's relevant to the master naturalist because it's a technique that can be used to answer questions about biodiversity. So I broached the subject with Lynn, and she thought it was a good idea to maybe have a little training session on PCR and what it is. And so that's what this is about. Um, okay, so when I talk to students, especially my graduate students, and they're talking about research, I always say, start with a question. So one question we can ask is, who lives here? How do we know, right? We want to know ecological type information about what are the organisms that are making up this part of the biosphere. And so how do you go about answering that question of who lives here? And I guess if we were in person, then I would just wait for you guys to answer my question, but it's a little harder in Zoom. But you can do like we do as a group. We go out when we're allowed to be together in person and do like bio blitzes, and we just see who's there. So we go around and we find the prairie dogs and monarchs and mallards, etc. And these organisms aren't too bad. We can go and use our field guides and see what matches, and we can count those, and we can sex those, and we can find all the information about them based on just morphology. So what do they look like? But sometimes gets complicated, right? So some organisms look the same. So are these the same or are these different? Are these the same or are they different? Are these the same or are they different? And so if you just go off of morphological information, then sometimes answering the question of who lives there gets more complicated, right? And I tell the class when we do the fungi for the training class that with all the fungi, a lot of them have asexually reproducing part and a sexually reproducing part, and the two parts don't look the same. So for a long time, taxonomists were calling the same organism with the different phases two different species, but they're not. It's the same species, it's just a different form. All right, so what if we want to say who lives in this environment, right? Especially like who lives in your gut? That's also an environment. Or you could say who lives in the prairie dog gut, or who lives on the prairie dog skin, or who lives in the ducks. That's one question we actually are asking at MSU. I have some students working on that project, and my wife has some students working on that project up in chemistry. The problem here is even more aggravated in that, so we have a human, we're like covered in this patina of microbes. So we have bacteria, we have viruses, we have fungi, we have protists all over us and on all of the mucosal surfaces. So your respiratory tract, your GI tract, everything. But if you take a sample and you grow them up, it's like, yeah, maybe those look kind of different, right? They're different color, but pretty much if you just go by what they look like, there's maybe three things there. And maybe there's a little more on this plate, but a lot of them look exactly the same. And even if you look at them under the microscope, they look the same. So are these two pictures of the same species, or are those two different species? So that sort of raises the question. If you can't rely on just what it looks like, then how can we tell if a given colony, let's say, is a separate species. Are all these the same organism, or are they all different? That's also a possibility. And then another complicating factor is that a lot of the organisms, especially the ones that live inside our gut, we don't know how to grow. So we can't even do this part. We can't even try to grow them up to a big enough size where we can see the colony. They're what we call non-cultured. But we now know that they're in there. So what other characteristic can we use to answer the question of who lives in a particular environment? All right, so I know you're all thinking DNA, right? We use DNA sequencing and we see who's related to whom, right? So building family trees, who's related to whom by blood, right? That's really just your genes, your genetics. And just to remind you a little bit, because we're going to use this part later, DNA is a double helix, and the important parts are what are called bases. So it's G, T, C, and A. Everything in your DNA is a G, a T, a C, or an A. And it's a double helix, so there's two strands, and the other strand is complementary. So G's always go with C, T's always go with A. Okay, so if I get to a point where I need to go over something again, then just yell at me. So I didn't want to go into like DNA structure in great detail, but I do think it's important to note A always goes with T, because this is going to be important later. G's always go with C's, and it's the sequence, what order the G's, T's, C's, and A's are in, that make the blueprint for the organism, right? So this is the code that codes for making a U, right? Or a code for making a flicker, or a code for making a mushroom, right? Everything that we need to make us is in this genetic code. All right, so we can do genetic sampling, and the process, when you summarize it, sounds really simple. So you just take some kind of sample and you do some preparation in the lab, and then you extract out the DNA, and then you put it in a sequencing machine and you get a sequence, and then you can analyze what you get out. And typically when we're talking about doing the things that we're talking about, which is who lives there and who's related to whom, we need to take the G's, C's, T's, and A's and then line them up, right? So here's, if you did 3 DNA sequence reactions, you might get these 3 sequences. And then you can use a computer, or you can do it by eye, and you say, all right, well, if we line up all the common ones, then we get this. All right, so we can see who's related to whom a little bit depending on how these guys line up. All right, so if organisms are really closely related, then their gene sequences will be really, really similar. All right, so this is an example of that. It's a very conserved protein, so that means lots of organisms have it, and it doesn't change very much over evolutionary time. So let's see if anybody will pop up with this. So how are these all related? Like, why do you suppose that there's so much commonality in this DNA sequence for these organisms?
All mammals?
Yes, they're all mammals. So you can see that even though we would say that there's a big difference between a mouse and a human, right? You could say, well, yeah, mice and rats are going to be pretty close together. And maybe humans and chimps we accept are close together, but really a human and a mouse? But no, we're all mammals, so all of these sequences are really, really similar. Now, if you say, okay, well, now we're going to compare a human to a pine tree, okay, well, then those probably won't line up as much. Those are going to be a lot different. So the idea is that you use the differences in the gene sequences, sort of these kinds of things here, and we can build trees. So we can say these organisms are really closely related. This is sort of a first cousin, and then these are not very closely related at all, right? So all of these kinds of trees that you might see are all based on analyzing how closely the sequences match to each other. Okay, are we good so far? All right. Okay, so when we are going out and sampling and trying to answer these questions of biodiversity, what kinds of samples could we collect? So I'm gonna go ahead and ask this again. So what do you guys think? Give me some ideas of the samples that we could use to figure out who's living somewhere.
Well, you could use a visual sample just by what you see there.
Well, true. So that helps with the macro fauna, but it doesn't help with the microbes. And then even doing a visual sampling, that's what we were talking about first. That doesn't give us the DNA. So if we wanted to actually collect DNA from different organisms, what would we need to get, do you think?
Blood.
Blood? Okay, so blood's a pretty good source. Blood's a little complicated, and I'll try to come back to that and explain why later.
Excrement.
Excrement, yes. Okay, so scat, right? So we'll go on, and so you could say getting any kind of tissue. The problem is that if you're going to do that, then you actually have to capture something, right? So even if you're going to draw blood, right, we would have to capture it, and then that requires permits, and that requires skill, and you're always worried about hurting the animal. So a lot of what the biodiversity people are doing is they will collect scat, and this works because as the fecal material passes through, it picks up epithelial cells from the lining of the intestine. That's how the Cologuard works for doing colon cancer screens. I just had my physical, that's why Cologuard's on my mind. But you can use this to collect DNA both from the animal itself, but also there may still be microbes inside the scat that you can get information about. Or you could just take water samples if you're interested in what's in the water column, or sediment from ponds and streams. But another common source of epithelial cells or tissue are hair traps, right? So this is, my sense is this basically is fancy barbed wire. And then as the animals pass under it or over it, trying to go down the path, then they snag their fur on the barbs, and you get this little sample of tissue, skin tissue, or hair follicles on the hair traps. Or people will go around and just look for rubbing spots. So this tree, it looks like that tree has some barbed wire on it as well, but just regular trees or rocks, you might get trapped hair samples. All right, so if you think about it, there's a problem of scale. All right, so I put in this blank to remind me to talk about this. So, if you're going to get a hair sample off of one of those hair traps, there's probably some cells there, but it's not huge, right? So we're talking, like this much tissue. And in the scat, most of it's going to be waste. And the number of animal cells in there is going to be pretty minute. So there's probably DNA there, but not very much. So part of the whole problem of doing this kind of sampling and trying to get at who lives here using DNA is, how do you get enough DNA to play with? Okay, so this is the problem that we're eventually going to try to deal with. Okay, so DNA is really cool, or it has a really cool characteristic in that it can make copies of itself. Right, this is what we need to do. Every time a cell divides, you have to make copies of the genome. You have to make copies of the DNA. And so I think I can put in here why blood is somewhat problematic. So you could not use, well, so if you're talking about humans, in the blood, what kind of cells do we mostly have?
Red.
Red blood cells, yes. Um, but if you were going to try to extract DNA from human red blood cells, it would never work because human red blood cells don't have a nucleus. So there's no DNA left in our red blood cells. There's white blood cells, and those have nuclei. So that's how you can use a blood sample for DNA stuff. And other animals have nucleated red blood cells, so you can use the red blood cells for that. All right, but so DNA's got this characteristic, it's this quality that it makes copies of itself. And the fact that it's two helices and A's always go with T's and G's always go with C's means that it's really good at copying itself. All right, and this process is called DNA replication. So here's sort of the short form version of what you need to make copies of DNA. So first is you have to have the DNA, which is this, and then it comes apart, and then you can make copies. And so you need all the extra pieces, right? So this, if you think about it, Jonathan's now off at college, so he's not playing with Legos anymore. But if you think of this as a big string of Legos, and you're going to make a copy of the string of Legos, then you need a whole bunch of extra Legos, right? So the A's, T's, C's, and G's that you have floating around the cell, those are like the pile of loose Legos that you're going to link into your strand here. And then you need a primer, which is a little piece of DNA that starts everything off. And then you need an enzyme called DNA polymerase. So this is just an enzyme. ACE is that indicates an enzyme, and it's making a polymer of DNA. All right, so if you have your old DNA, you have a bunch of ACs, T's, and G's, you have a little primer, and you have the polymerase, then you're good to go. You can copy your DNA. Okay. So in 1983, a guy named Kerry Mullis and other people in his lab were working at the Cetus Corporation, which is a private like biotech company. And they were mulling over this problem of, well, how do we take a little bit of DNA and turn it into a lot? How do we get DNA to replicate itself for us on purpose? And he developed this idea of saying, okay, well, we could have our template DNA, that's the old stuff, right? And we put that in a test tube, and then we put in a primer that we can build, and we put in all the A, T, C's, and G's, and we put in DNA polymerase, and we should be good to go, right? And so we just have to get the DNA apart, the two strands apart, like it does in the cell. And then the DNA polymerase will make copies of the originals, just like it does in the cell, but we're going to keep going. We're not going to split the cells. There is no cell. It's all happening in a test tube. So 1 becomes 2, 2 become 4, 4 become 8. And if you do it long enough, you go from 1 copy of DNA to a billion copies of DNA, and we can certainly play with a billion copies of DNA. All right, so this idea of how you would use DNA polymerase, this enzyme that copies DNA, to go from one strand or one chunk of DNA to a billion chunks of DNA. He said this was reminiscent of what happens in some atoms, right? So here you have a neutron hits this atom, causes it to split. That leads some neutrons, cause other atoms to split, right? So this is called a nuclear chain reaction, right? Which if you don't control it, turns into an atomic bomb. But this pattern of 1 becomes 2, becomes 4, becomes 8 was reminiscent of this. So Mullis called his DNA thing a polymerase chain reaction, or PCR. So that's where the name comes from. All right, so all PCR is doing is using DNA polymerase to take one double helix of DNA and turn it into a billion copies. But there's a couple problems or a couple challenges. So the first is you have to get the two strands apart if you're going to make a copy. And there's several ways you can do that. In the original cell, there's an enzyme here that does it. But that's another piece that you would have to put into your test tube, and you're trying to make this as simple as possible. So you can also get DNA to come apart if you use heat. So if you take DNA and you heat it up a little bit, then it unwinds and it comes apart. And then if you cool it, it will come back together. All right, so it denatures and renatures if you change the temperature. But DNA is not the only thing that's in the, so if you take your double-stranded DNA and you heat it, it comes apart. If you cool it down a little bit, then you let the primer sit down, and then DNA polymerase can make the copies. And then you just do it again. That's the chain reaction part. But getting the two strands to come apart, you have to heat it up to like 90 degrees C, which is close to boiling. And there's a problem that DNA is not the only thing in your test tube. You have DNA, but you also have this enzyme, which is a protein, and proteins also denature. So if you denature a protein, it comes apart, and then when you cool it back down, it doesn't fold up correctly. It won't work. So the analogy that we have for that is this. All right, so eggs are full of protein. If you heat them up, they denature and you get this. When you cool it back down, you don't get the original egg. Right? You can't unscramble an egg. You can't unfry an egg. So it will never go back to being that. Once it's denatured, then it's dead. So in his original experiments, when he was trying this method out, Mullis would have to add new polymerase every time. And that got really labor intensive, and it was really expensive. So it was going to be problematic. So PCR, as we know it today, depended on two advances or two discoveries. One was natural and one was engineering. All right, so the natural one was they found, and this again was people working for CETUS. They went out looking for useful enzymes in environmental organisms. So in this hot springs in Yellowstone called Mushroom Pool, people isolated this guy over here, Thermus aquaticus. And they found, and since this has been adapted to living in these hot springs, and the hot springs are like 95, 96 degrees centigrade, they must be able to stay alive. They must be able to make new cells, so they must be able to reproduce and replicate their DNA. So when the DNA polymerase was isolated from Thermus aquaticus, they found, as expected, that you could heat this one up and it wouldn't denature. It wouldn't become a fried egg. It would just continue to work. All right, so this is what we call TAC. So if you see the term TAC polymerase, that just means DNA polymerase, the enzyme that copies DNA from Thermus aquaticus. So they took the T and the AQ and they pushed them together. That's TAC. So we now had an enzyme that you could heat it up to 95 degrees and it wouldn't fall apart. It would stay active. And then the engineering fix, or the engineering advance, was development of a thermocycler. All right, so not these, this is one way of looking at thermal cyclers, right? But this isn't what I mean. It's one of these. All right, and the advance here is that they engineered an instrument that could rapidly change temperature. So you could go from 95 or 94 down to 55 and back to 72, and then back to 94, down to 55, up to 72, up to 94. So you would just keep going through these cycling, and this instrument is able to heat and cool within milliseconds. So this really rapid heating and cooling that allowed the cycling to be done automatically. And with the Taq polymerase, then you didn't have to keep adding Taq at every step. So again, when the original setup, when the method was being developed, Mullis's lab people had a bunch of water baths, and they just took the test tube and they moved it from water bath to water bath, and that with a stopwatch. And it always took a while for it to get to the right temperature. All right, so with those two things, we can actually do this now. And like all engineering, or like most engineering things, the cost of the thermocyclers has come down drastically. So now this is relatively affordable for any lab that wants to do it. All right, so just to give you a couple of examples where this is actually being used. So here's a paper that I pulled from Conservation Genetics Resources. So they're using PCR, amplifying genes that are specific for bats to identify the bats just from their scat, just from the bat fecal samples. And then this was a paper on the bison herd out in the Panhandle, I think. And they're using this to keep track of who's related to whom, so they don't have a lot, as much inbreeding, so they can keep the stock genetically diverse and keep them from having medical problems because they're too genetically identical. All right, so I wanted to add one more little piece in, and that is a modification. So what we talked about so far was just the classic PCR, using that enzyme DNA polymerase to turn one or a few pieces of DNA into billions, and then we can analyze them. We can do the sequencing, we can then get the sequences aligned, and we can figure out who's related to who, we can identify who's in a particular environment. But there's another way of looking at this. So people started to say, well, I want to know what's there like right now. I want to look at this amplification of DNA in real time as it's happening. And so the modification to the technique is they've added in a molecule that fluoresces, so it lights up when it is sort of meshed into the DNA double helix, right? So when you have this molecule that's sort of sitting inside the DNA double helix, then it fluoresces, it gives you a light. When you go through the PCR reaction and the two strands separate, then the molecule comes out, it's floating around, and it doesn't light up anymore. And then after you make your copies, now you have double-stranded DNA, the molecule goes in there and lights up, right? So in the instrument, as you're watching this happen, if there's one or a few or five double helices, when the DNA number is low, then the signal is low. When the DNA number is really high, then the signal is high. So the more DNA, the more light is produced, or more fluorescence is produced. All right, so that gives you this. So when you put it in the instrument, it comes up on a display. The instrument's just detecting the fluorescence. And there's a lot of considerations that go into what your threshold is. That would be a talk for another time. But you can see you start with a few copies of DNA, a few pieces of DNA, and the signal's really low. So it's like it's really dim, it's dim, it's dim, and then your numbers start to build. And at some point, you say, okay, this is bright enough, I'm going to call it positive. All right, and then it'll keep going and eventually plateau because the camera gets saturated. So the cycle at which it crosses threshold is called the CT number. All right. And so one of the last thing I wanted to explain about this is if you have fewer pieces of DNA at the beginning, then it's going to take more cycles to cross the threshold. If you have a lot of DNA present, then it's going to take fewer cycles to get through threshold, right? So if you started with one and we can detect, say, 10,000, if you start with one, it's going to take a whole bunch of these cycles to get up to 10,000. If you started with 5,000, it's only going to take you one cycle to get to 10,000, right? So that's what this graph's supposed to show. So if you have a few copies, like just 10 copies of your DNA, then it will take you a long time to get up to threshold. If you have 10,000 copies of your DNA, then you cross the threshold a lot sooner. So in this case, your CT number might be 35, and here your CT number might be 25. Right. So when you're talking about this kind of PCR, um, you want your CT number means there's more DNA there, more or less DNA there. All right. And I bring this up because this has been in the news lately, or in some of the science stories about detecting using PCR to detect SARS-CoV-2 and talking about CT scores. Okay. So I don't know. That feels like I went through it kind of fast. So, but that's what I had for today. So I'd be glad to answer any questions or go over something again that wasn't clear.